Okay, thanks, Gary. Okay, so one of the things I've always really enjoyed about being at Vanderbilt was my collaborators, of which I've had a lot of people. Seems not to be working, just another thing that's not working. Okay, well, that's interesting. Okay, now why is that not working? It was working a second ago. Okay, there we go. Let's see if it's going now. So these are some of my former collaborators at Vanderbilt, of which some of them are still here, and so these are some of my current collaborators. And if any of you are collaborating with me and I didn't get you on the slide, I apologize. I have a few senior moments now and again, but that's one of the things I've always really enjoyed about being here, is our collaborators are working together. Okay, and predominantly the person who just invited to this particular lecture is Gary here with his zombie friend, and I'll get to the part that Gary really wants to hear near the end. So we do a lot of different things in the lab. We look on fundamental clock mechanisms in organisms from bacteria to humans, adaptive fitness and evolution of clocks, clock properties in Angelman syndrome model mice, and development of luminescence techniques. And the common threads are clocks and luminescence recorders. What I'm going to talk about today is what are circadian clocks good for, how do they work in bacteria, especially adaptive fitness and mechanism, and drugging the mammalian clock, a redox. Okay? So circadian rhythms adapt to a cyclic environment. I've got this nice video of a sunflower, a heliotropic sunflower, during the day, tracking the sun and the movement of the sun during the day. And what's cool about it is not only does it tracking the sunlight as the sun moves across the horizon, but as we go into night, what happens is our circadian clock says the sun's going to come up in the opposite direction for dawn, and it now resets the heliotropic sunflower so that it's ready for sunlight dawn the next day. And that means then that of these things that we often think in terms of homeostasis, actually the set point for homeostasis is not constant, but it's actually an oscillating set point, especially for hormones and many of these other different things that are the daily cycle of circadian physiology in humans: sleep, metabolism, gene expression, hormones, insulin sensitivity, athletic performance, alertness, memory and learning are all things that are controlled and influenced by a circadian clock, so that these things are changing as a function of the time of day. So what does your circadian system do? Well, there's anticipation. There's the kind of a program that anticipates changes in the environment and makes sure that things happen appropriate for that. But there's also a continuously consultable clock, because you have a feeling for what the time of day is. And then in the case of birds and bees, they actually use that for orientation. Our circadian clock is also how we measure the day length, and therefore elicits the different seasonal responses, which are very important in reproduction and other things. Okay, so one of the programs that we often think about is avoiding predation. And usually people will say, well, you know, these cute little nocturnal rodents that go down into a burrow during the day to avoid the day active predators, and they come out at night, so there's not as many predators. But let me show you a different kind of a case in plants, where plants that anticipate rhythmically active herbivores also mount their defenses rhythmically, such as putting secondary compounds in their vacuoles and whatnot, but these things have to be phased properly. So what I'm going to show you in this video is a cabbage leaf here and a cabbage leaf here, and caterpillars are put onto the cabbage leaf. And in this case, the cabbage leaf and the caterpillars came from the same light dark cycle. They're in phase with each other, and the plants can't anticipate that. But over here, they were in reverse light dark cycles, so they're not in the same phase, and the plants aren't able to anticipate very well. So here we are, we've got the cabbage leaves, and the caterpillars are on them, and they're chomping down on them. And as we can see, as it progresses, the cabbage leaf that's anticipating the herbivores has mounted a more effective defense than the cabbage leaf that's out of phase with it, and it gets eaten up and chomped down a lot faster. Okay? So the phasing of these things is important when we're thinking about avoiding predation, even in a case like this. Okay? So I've got, I often use a lecture title as time blows by, because we use a lot of luminescence reporters. And I'm going to talk today about using luminescence reporters to assess adaptive significance and the evolution of rhythmicity in bacteria, discovered and analyzing this nanomachine clockwork that I'm going to talk about. And then also with Gary, discovering new clock-active drugs for people, okay? So one of the reasons why we study Cyanobacterium synechococcus, here's a picture of them, basically green E. coli, is they have a lot of technical advantages, especially genetic and whatnot, and we developed this luminescence reporter system, but they're also globally and evolutionarily very important because of the oxygen that they produce. Evolutionarily, they're responsible for our having an oxygenation and an ozone layer and that sort of thing that enabled humans to come to be, and they're still doing that globally, and biotechnologically, we and other people are using them to make bioproducts that are useful. So our luminescence system in both bacteria, and the same thing I'm going to talk about in mammals later on, is basically using the clock controlling a promoter that causes the transcription and translation of a luciferase enzyme that produces light, and that since the synthesis of this is under the control of the clock and it gets degraded when the clock's not turning it on, we can get rhythms of luminescence over the day that are these beautifully precise readouts of gene expression and the promoter activity. And what's being shown here are two different cultures that had been trained to light-dark cycles that were out of phase with each other. This was on a light-dark cycle. This is on a dark, light, so this is reversed light cycles. And you see this beautiful continuous rhythm in constant conditions, but it had been phased in opposite phases by the prior light-dark cycle. Okay, so that luminescence system led us and other people to undertake mutagenesis screens that ultimately identified 3 key clock genes in cyanobacteria, called the Chi-A, Chi-B, and Chi-C genes, where we have wild type. If we knock out any one of those genes separately or the whole complex, they become arrhythmic. Okay, and these homologs are found widely through bacteria, both archaebacteria and eubacteria. So, do these clocks actually do anything useful? Well, we got onto this, and I'm going to tell you probably the experiment that my laboratory is most famous for, and that is to use a test that evolutionary biologists and population biologists often use to assess adaptive fitness, and that's the competition assay. Okay? So the idea is to compete different strains against each other to see what properties allow adaptation to environmental conditions. So we could take 2 different strains of cyanobacteria, say one that has a fast clock and one that has a slow clock, mix them together, grow them in competition under different environmental conditions, and see basically who wins, okay? Who outcompetes the other. And so this experiment is one where we competed wild type with period mutant strains. So we had a wild type strain, we had one that had a clock mutation that caused a fast free running period of about 23 hours, and another mutation that had a slow clock of about 30 hours, okay? Whereas wild type is usually about 24 or 25 hours. And what we did is we mixed those together and competed them in light-dark cycles of either 22 hours, so light-dark, 11 hours of light, 11 hours of darkness, or a 30-hour cycle, 15 hours of light, 15 hours of darkness. Notice that over time, the amount of light and dark is equal. It's a photosynthetic organism, light's important to it, okay? But the frequency is what's different, all right? If we compete the short period strain against the wild type, and mix them together so they're about 50-50, then split that culture, put half of them in the 22-hour light-dark cycle, and the other half in the 30-hour light-dark cycle. What we see is that after about a month, the short period mutant has taken over the culture. After a month in the long period light-dark cycle, the wild type's taken over the culture, right? If we do the competition of wild type with the slow clock, the long period mutant, start out at about 60-40, in the short period light-dark cycle, the wild type's taken over the culture, but in the long period light-dark cycle, the long period mutant has taken over the culture. And if we compete the short period mutant against the long period mutant, we get the very clear result that the short period mutant wins in the short period light-dark cycle, the long period mutant wins in the long period light-dark cycle. So that means that having a free running period that's constant or resonates with the environmental light-dark cycle is adaptive, okay? And so the conclusions from those fitness tests in cyanobacteria is that strains that have a period similar to the light-dark cycle outcompete other strains, and persuasively, the mutants can outcompete the wild type, which I think is really cool, okay? It's not so surprising that wild type will defeat a mutant, but to have the mutant defeat the wild type in the environmental condition that's appropriate for it, that's what makes this experiment persuasive, okay? And therefore, having a clock with a period that matches the environmental cycle enhances fitness. And that's true for both intraspecific competitions, which is what I just told you about, but we have unpublished data where we're now competing different species of cyanobacteria, so even interspecific competition, that same result holds. So the clock system confers an advantage towards adaptation to rhythmic environments. So now let's talk about this nanomachine clockwork mechanism. Okay, well, in higher organisms, we talk about the transcription, translation, feedback loop mechanism for circadian clocks. The idea that there's a rhythm of transcription and translation of key clock genes and blah, 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 okay? In cyanobacteria, we have a transcription, translation, feedback that globally controls gene expression, but really interestingly, there is a core clock that is strictly biochemical, which is what I'm going to talk about now. So this whole clock system is controlling global gene expression, it's controlling chromosomal topology and condensation, the time to cell division, nitrogen fixation, and this competitive advantage and fitness thing I just told you about. And so now let's talk about how this thing is put together. Well, it's cool that in cyanobacteria, all three of these key proteins that I told you about from those key genes, Chi, A, D, and C, they've all been crystallized. We know the three-dimensional structure of all three of these key proteins. The Chi A gene is, Chi A protein is a dimer of about 30 kilodalton monomer, and that was done by other labs. Chi B is a tetramer of a very small protein, 11 kilodaltons. And then with Martin Agley, who did 95% of the work, we purified the protein form. We determined the structure of the largest protein, the hexamer of Chi C, which is this hexamer, double domain hexamer that we're going to talk about. I love this quote from Jacques Monod in his Nobel laureate speech, that the ambition of molecular biology is to interpret the essential properties of organisms in terms of their molecular structures. I think that's what molecular biology is all about. And in the case of the cyanobacterial clock mechanism, we're really close to fulfilling that ambition. Okay, so let me talk about that. So one of the things that was noted fairly early on in the study was that this big protein Chi C gets phosphorylated and dephosphorylated on a daily cycle. Well, our collaborators and later competitors in Japan, Dr. Kondo's lab, did an incredible experiment, and they purified those three proteins. They put them together in a test tube, and they added ATP, and they found that they could reconstitute the rhythm of Chi C phosphorylation just with three proteins in a test tube. And what we're seeing here is a gel from taking out from that test tube for 3 days, where we have the hyperphosphorylated Chi C, the hypophosphorylated Chi C, hyperphosphorylated. Conveniently, they run a little bit differently in STS polyacrylamide gels. And this is actually showing the rhythm for 10 days, and I'll show you an even more fantastic picture in a minute. But it's even temperature compensated in vitro. That is that the period is at about the same period at different temperatures, which is one of the key characteristics of circadian clocks. Just so that you really get this, okay, this is an animation my former graduate student did. Here's these three proteins just in that test tube with the ATP, and then over time the Chi C hexamer gets phosphorylated, and then gets dephosphorylated, and then gets phosphorylated, etc. And that's what the gel is right there, okay? So, let me now summarize work from a lot of different laboratories, not just our laboratory, on what we think is going on in that cycle. We started with the Chi C hexamer here. There's interaction of Chi A at the C-terminal tentacles of Chi C, okay? So it comes down and interacts, and I'm going to show you that with AFM in just a minute. And that interaction leads to the phosphorylation inside Chi C, first at a threonine residue, and later at a serine residue, so we get a hyperphosphorylated Chi C. That hyperphosphorylated Chi C now can interact with this small molecule, the Chi B, which then recruits the Chi A down to this region, and this is no longer stimulating the phosphorylation, so it starts to dephosphorylate, first off the threonine, and secondly off the serine. And so we have the phosphorylation cycle, the dephosphorylation cycle, and the formation of a stable complex of the three proteins during the dephosphorylation phase. During the phosphorylation phase, there's a labile interaction between the Chi-A and the Chi-C, leading to the Chi-C phosphorylation. So I want to talk about this labile interaction between Chi-A and Chi-C and tell you what we learned with AFM. So Chi-A interacts with Chi-C and facilitates the Chi-C autophosphorylation. And we wondered if Chi-C's phosphostatus, that is, whether it was phosphorylated or not phosphorylated, could feed back on its interaction with Chi-A, thereby modulating its own rate of phosphorylation. So here's the Chi-C, the Chi-A interacting with it. Does this interaction, does it change the kinetics of the Chi-A, Chi-C interaction? This is actually negative staining images of Chi-C with Chi-A interacting with it by EM. And so we're going to use AFM, atomic force microscopy, to look at that interaction. We're going to stick Chi-C down onto a mica slip, and we're going to look to see whether Chi-A interacts with it by the cantilever. So atomic force microscopy may be familiar to you. This is high-speed atomic force microscopy that we did in collaboration with our Japanese collaborators, Ando Sensei and Uchihashi Sensei. And the idea is that you stick molecules down on a surface, and this cantilever goes and scans what are the peaks and valleys, okay, what's sticking up and what's not sticking up, and you can scan through and start to get a picture of what's on the surface, okay, where we're using Chi-C stuck down onto mica and get a picture of that. And because it's now being able to do that, so scan so quickly, we can really look at kinetics of molecular interaction. Okay, so here's a picture now of each of these orange blobs right here is a Chi-C hexamer, okay? And you'll notice a kind of a bright spot that occasionally occurs. That's when Chi-A binds onto it, so it makes it higher. So as the cantilever goes and scans it, says, oh, there's something higher there. So this is a Chi-A interacting with one of those Chi-C hexamers, okay? At higher magnification, we can also see these Chi-C hexamers and then the bright spots with the Chi-A binding onto it. This is approximately in real time, and you can see it doesn't last very long, maybe a second, okay, our interaction. So it's a labile interaction. So the question we wanted to ask was, does Chi-C's phosphostatus influence the kinetics of that reaction? So over here we have a sea of Chi-C hexamers that are pretty much dephosphorylated, and another sea of them over here that are pretty much phosphorylated. Chi-A has been added over here. Now you see the bright spots, okay? And now I'm going to add Chi-A over here, okay? And you see the bright spots. And it may take a while to get used to this, but if you look at it carefully, you see that this looks like it's flickering faster than over here, okay? What that means is that there's a shorter dwell time with the Chi-A on Chi-C over here with the phosphorylated hexamers, whereas with the unphosphorylated we have a longer dwell time, higher affinity and lower affinity. So the Chi-C phosphostatus appears to be influencing how long the Chi-A will interact with. So this is an experiment done by my research associate, Dr. Mori, where basically he did for over 2 days straight. He went in and every 4 hours he took out a sample from the test tube. He stuck it down on the grid and they made the measurement. You know, they just did a straight 2.5-day experiment, basically. No sleep, okay? And what they found, here's the rhythm of the Chi-C phosphorylation I've been telling you about. But now if you look at the binding lifetime is pretty much in antiphase to that. So that when the Chi-C is hyperphosphorylated, the binding lifetime is low. When it's dephosphorylated, the binding lifetime is high. That's what's being shown here. So this is at hour 15. This is the trough of the phosphorylation. So hypophosphorylated, the dwell time is relatively longer than at hour 24 right here, where now the binding lifetime is very fast, and the Chi-C is hyperphosphorylated. Okay, so we get this beautiful curve here of relationship. Chi-C phosphorylation, high phosphorylation is fast binding, okay, fast on off, whereas if it's hypophosphorylated, the binding lifetime is longer. Okay, so what? Okay, well, it turns out we did simulations on that. So Chi-A binds strongly to the trough phase hypophosphorylated Chi-C. It binds weakly to the peak hyperphosphorylated dwell time, and so we started to do a lot of simulations of this. And we found that this phenomenon, this sort of phosphorylation-dependent differential affinity, increased the range of the allowed Chi-A and Chi-C stoichiometries, okay? This is important because in the intracellular milieu, there may be times where the stoichiometries of the proteins are not all going to be exactly the same, okay? So you want to have a range of stoichiometries in which this oscillation can occur. And so what we did here is in the simulation, if you look at the ratio here of Chi-A to Chi-C, that with this phenomenon, which is the usual phenomenon, the phosphorylation-dependent differential affinity, now we have a much larger range in which oscillations can occur, and that the period is pretty much conserved at the base, at the regular level that we're preferring. Whereas in simulations, we can take that property out of the system, and now the range of allowed oscillations is much narrower, and also the periods start to really dramatically increase, and so we're not conserving our period that we'd like in the absence of that phenomenon. So this non-equilibrium phosphorylation-dependent differential affinity broadens the range of Chi protein stoichiometries that allow rhythmicity. It refines the period and conserves the period of the 24-hour oscillations, okay? So I think what's cool about this is that the 24-hour time course of the resilience of the oscillation is partially explainable by molecular events that are occurring on the time scale of subseconds, okay? And so the idea is that we have the Chi-C phosphorylation rhythm that early on in the dephosphorylation and just beginning of phosphorylation, the dwell time is relatively long, but as we get higher and higher and more phosphorylated, the dwell time gets shorter and shorter. The way we can understand this intuitively for a dummy like me, okay, is that if we have a one hexamer that's going up this curve faster than the rest of the population, as it gets up here, the dwell time with Chi-A gets shorter, and that probably reduces the probability that the interaction of Chi-A will lead to a phosphorylation event. So the rate of phosphorylation may be fast here, but it's going to slow down here. Whereas a hexamer that takes off a little slower and is kind of behind the pack after a while, but because of this phenomenon, the rate will be fast here, and the fast guys will be slowing down. So basically, it keeps the pack of hexamers pretty much in concert with each other, and so that they can reach the peak at about the same time. Okay, so that's what we were talking about here, that this is the inside biochemical oscillator, and that the rhythms that are known for this are the phosphorylation status of Chi-C and ATPase activity of Chi-C, conformational changes, formations of complexes, and also monomer changes that I didn't mention. And then this is now regulate the transcription translation feedback loop. So now let's talk about this complex that's forming, the Chi-ADC complex. This is the rhythm of phosphorylation. Way back in 2007, we showed for pretty much the first time using a negative stain EM that these proteins were forming a complex, okay? The Chi-ADC complex and then just a regular Chi-C hexamer, that during the phosphorylation rhythm, in the phosphorylation phase, it's pretty much free Chi-C and with some AC interaction. But in the dephosphorylation phase, we now have these larger complexes forming, which are fairly stable as this stable complex. Okay? Well, that's led to a new and automated assay that we can do of this oscillator. So in the phosphorylation phase, pretty much these proteins are free of each other, especially the Chi-B is just floating around free. But in the dephosphorylation phase, they form this stable complex. Chi-B is small, it's only 11 kilodaltons. So if we put a fluorophore on that, we can look at kind of the rotation of the Chi-B by fluorescence anisotropy. And the idea is that if we put a polarized excitation then if there's a rotation of the molecule during the fluorescence lifetime, we can depolarize the light and look at the fluorescence anisotropy, okay? In this case, Chi-B is still in small, as it's small protein, just 11 kilodaltons, so it may be rotating relatively fast, but over here, it's now in this much larger complex of about 500 kilodaltons, rotates much more slowly, so fluorescence anisotropy can be used to make an automated assay of the rhythm. And in fact, that was shown by the laboratories of Andy Lang and Michael Russ, where here's the anisotropy. This is for 7 days. There's this beautiful rhythm. basically of the formation of the complex, and then the complex falling apart, and the complex coming back together again. We've also adapted this assay now, and this is data from Dr. Mori again. This is 1 month, okay? These are the rhythms of the fluorescence anisotropy of 3 proteins in a test tube with ATP for 1 month. It just, blows my mind. Then he put it in the refrigerator for 298 days, okay? Then he put it back at 30 degrees, and it's still oscillating, okay, after it warms up again. It's just like, wow, I can't believe it, okay? It's just amazing, okay? And we're using this assay now to analyze some mutants of Chi-C that are defective in the key property of temperature compensation. Okay, so I alluded to the fact that this clockwork that we've been talking about controls global gene expression, and those links have now been made. It turns out that the phosphorylation status of Chi-C influences linkage kinases SAS and KIK that then cause a rhythm of the phosphorylation of a global transcriptional factor called RPAA, which then binds the target promoters and elaborates the global gene expression. The laboratories of Dr. Luang and Susan Goldin have just published a paper in Science where they now put the fluorescent fluorophore on all of those proteins, either on Chi-A, or they put on Chi-B, or put on SAS, or KIK, or RPA, and they basically show by fluorescence anisotropy that all of those things are coming together in a complex, okay? So that when Chi-C is phosphorylated, it's recruiting all of these things into this complex, including the global transcriptional factor, and then even the DNA of those target promoters is binding to that complex, and it's also showing rhythms that you can detect with fluorescence anisotropy. So pretty much that entire system of the output has also been reconstituted in an in vitro assay. Okay, so what are some of these outstanding questions with the cyanobacteria? Well, evolution. The Chi-C-based time-keeping systems in cyanobacteria and other bacteria confer an adaptive advantage to organisms in a cyclic environment and suggest the selective forces that led to the evolution of these timekeepers. The linkage between the Chi-C phosphostatus and the TTFL, that's the transcription correlation feedback, may be understood, but how does this 3-protein nano-timekeeping machine really keep time? Is it the association-dissociation of the complex, which is what I've been talking about so far, or is it that there's some constant rate of something that gets converted into a cycle, kind of like this water clock that I videotaped in China. There's a constant water flow, but because of the configuration of this system, it can now be converted into an oscillation. And in the case of Chi-C, what we believe is that there's a constant low rate of the ATPase activity, okay, that then leads to a tension in the molecules, leads to the complex forming and unforming, as I mentioned, but the complex forming and unforming is not the core rate. It's probably the ATPase activity, and that the complex formations are what kind of convert all of this into a cycle, just like this part converts the constant stream into a cycle, okay? And that's still, how that happens is still a completely question outstanding out there. Okay, now let me talk about looking for clock-active drugs in people, and this is where Gary kind of reiterates the story. Okay, so here's some of the consequences of having a clock for you and me, okay? We have shift work, jet lag, we have sleep disorders, we have impaired performance and alertness, such as these guys on parade where they just became, they got zonked out. Chronopharmacology refers to the fact that because our circadian physiology is rhythmic, that also the efficacy of drugs is often rhythmic, okay? Those of you who take statins, as I do, for cholesterol, the optimal time to take it is in the evening because the statins are trying to knock down an enzyme in the liver which is strongly expressed in the middle of the night. So the best time to take the statin is before you go to bed to knock down that nocturnal peak. Affective disorders, especially seasonal affective disorder, but other depression and mood disorders are affected by the clock. Shift workers are especially prone to a number of different kind of health problems, cancer, gastrointestinal, cardiovascular problems. Metabolic disorders, one of the hot things in the field of circadian rhythms right now is that it's not only how much food you eat and how much exercise you get, but the timing of when you eat is important important because your circadian clock is regulating your metabolic rates, okay? And that's why it's good to try to not eat after supper, to allow a good fast so that you go into fat burning mode optimally when you're sleeping. Accidents and alertness affect medical personnel on call and also people that fly our airplanes. Okay, so can we get a jet lag pill? Because shift happens, okay? Could we get a pill that would phase the shift of the clock but have no side effects? And you know, jet lag is one application, shift work is another one. Okay, this is a homeopathic thing that's just a joke here for me. The only really currently available drugs for this is melatonin or memeltian, which is a melatonin receptor agonist, but the pharmacokinetics are poor because basically they get cleared out so quickly, it may help you to get to sleep, but it doesn't keep you asleep. And so it's not really very helpful for jet lag. High-throughput screens to discover new clock-active drugs have been successful, but have mostly found clock-active compounds that target generic kinases and/or transcription translation, and therefore have a lot of undesirable side effects. Okay, so we and other people have used luciferase reporters to try to undertake high-throughput screens. Okay, and we use a luciferase reporter in mammalian fibroblasts. Here's a rhythm of luminescence from these transfected cells. These are human U2S cells, okay, showing this beautiful daily rhythm, okay? And what we did is we talked Dave Weaver in the high-throughput screen facility 15 years ago into letting us use the high-throughput screen facility over the weekend for 3 days, okay, where we basically put them in on Friday and we take them out on Monday, okay? And look at the rhythms of cells in 384 well plates, okay? So our primary screen was testing about 7,000 compounds in each experiment using 20 plates by 320 compounds for these 3 days, and we succeeded in getting about 82,000 compounds screened through, okay? The phenotypes included short period, long period, phase shift, low amplitude, but how to prioritize and analyze, and these are just a few of the samples from those 3-day experiments. Well, this re-enters Gary here, okay? Gary noticed an interesting chemistry among some of the most potent long-period inducing compounds, namely that they had a phenazine group, okay? Which is this structure right here, okay? And this is one of our lead compounds right there, okay? And it turns out that at different doses, that this particular phenazine has a pretty dramatic effect on lengthening the circadian period from a control value down to 25 micromolar, 33. So that's actually a 50% increase in the free running period. Well, phenazines are interesting because this group can act as an electron shuttle, okay? So it can be reduced by FADH2 or NADH to a reduced form, and then oxygen can reoxidize it to the original form. So we can have this electron exchange and shuttle. So that brings us to this picture right here. So they can act as electron shuttles, and this is actually a population of bacteria in a test tube that when, with a phenazine, that when it's oxidized, it's green. But as it gets reduced, it turns to an amber color, okay? So there's bacteria in here and a phenazine that can shuttle back and forth across the membrane. And what's happening is, here's the bacteria. In the cell, the oxidized green form of the phenazine can get reduced by NADH, okay? Then that can move across the membrane and basically have the reduced form in the extracellular medium, but then in the presence of oxygen, it can get reoxidized and turn green and shuttle back and forth, and that's what this person's doing. As they vortex it, they're reintroducing oxygen, goes back to the oxidized form of green, then goes back into the bacteria, gets reduced, turns amber, blah, blah, blah. Okay, so that's just a visualization of how they can act as an electron shuttle, and thereby potentially affect the intracellular redox, okay? So, does that really happen in the mammalian cells? Well, re-enter and to the rescue Dave Piston, who used to be here, who has this cool technique of looking at NADH and FAD because they're both endogenously fluorescent, and by microscopy at their appropriate wavelengths, you can look to see how much of NADH and how much of FAD is present. And that's what these are. These are cells, okay, that are being excited, and we're looking at the fluorescence. And this can be used then to calculate something called the optical redox ratio, okay, and that's what's being plotted right here, okay. So we have NADH, FAD levels, and it turns out that these are the controls here, and this compound I'm just telling you about
changes and reduces NADH, but it increases FAD, and this is a control phenazine-like compound, but can't actually do electron shuttling. And that means that then the optical redox ratio with this compound is dramatically increased, and so it's more oxidized, okay? And basically that ratio quantifies the relative rates of cellular glycolysis and oxidative phosphorylation. So it turns out that that phenazine that we found in that screen does change the intracellular redox in mammalian cells, okay? So they slowed the circadian clock, changed the intracellular redox. Perhaps this particular compound had too many side effects to use as a pharmaceutical in humans. Okay, I'm not sure I really want to be changing my redox all over the place just for jet lag. But this compound now could be potentially a really useful research tool, not only for circadian studies, but for anything else where you want to change redox, okay? Phenazines are produced by bacteria, including gut bacteria. Could it be possible that your microbiome right now is releasing phenazines in your gut and affecting your circadian clocks itself? So that's a scary thought. So here's the take-home messages. The circadian timekeeping systems confer an adaptive advantage to organisms in cyclic environments. The central quartz crystal pacemaker, actually I just want to have this brief tangent here. With my undergraduates, I one time mentioned something about a quartz watch, okay? The quartz crystal watch. And nobody knew what the heck I was talking about. And I thought, wow, am I that far out of date? Because I go back to way back when quartz crystal, that was a cool thing. Okay, so the central quartz crystal pacemaker of the cyanobacterial circadian system is a 3-protein nanotimekeeping machine that can be reconstituted and studied in vitro, including single molecule analyses of resilience, which is what I talked about today. Luminescence report allow high-throughput screening for compounds that might be useful as clock and or sleep medications. And here's the people that actually did the work in my lab, Cyanobacteria, Dr. Xu, Dr. Mori, Louisa Jabur, Dr. Byrne is our mathematical simulation guy. In my lab, the people that were involved in the high-throughput screening was Dr. Hugo Bersetti, who did the screen 15 years ago, and more recently, my former graduate student, Dr. Kevin Kelly. Our collaborators on today's project were the people in Japan with the atomic force microscopy, and then Gary and Kwang Ho Kim here in the VICB. Dave Piston, who used to be here, but it's not here anymore, unfortunately. And then Hassan Musharraf, with whom I enjoy going out to have lunch and talking science and then arguing about politics. Okay, so always looking for motivated postdocs and graduate students, got some open positions along those lines. And I want to thank the National Institute of Health for funding, and especially grateful to those of you who reviewed our grants and manuscripts, and please follow us on Instagram. So with that, I'm finished with 1 minute remaining. Anybody got a question?
There's some questions on it.
Oh, okay. That means I have to do something, right?
Start pushing buttons.
Yeah.
You might be able to read them, right?
Okay, well, do I have to stop share to do that?
I think Q&A.
Oh, I can go to Q&A. Okay, I see one. Okay, is the kinase and phosphatase activity internal to Chi-C, and if not, has it been specifically shown that A is the kinase and B is the phosphatase? Okay, that's a great question. All right, so the answer is that Chi-C can autophosphorylate itself and auto-desphosphorylate itself without Chi-A or Chi-B ever being involved. How do we know that? If we take Chi-C and put it in the refrigerator at 4 degrees, what will happen is it will slowly phosphorylate itself without any Chi-A ever being around, okay? On the other hand, if we put it at 30 degrees without Chi-A, it will slowly dephosphorylate itself. So Chi-C is able to both do the phosphorylation reaction and the dephosphorylation reaction in itself without any other molecule being involved. And what we believe is that the rates of those two competing reactions are differentially temperature dependent. So when you make it cold enough, the phosphatase is inhibited a little more than the kinase, and so you slowly phosphorylate. And then when you bring it back to 30 degrees, now the phosphatase reaction is a little faster than the kinase reaction and slowly dephosphorylates. What Chi-A does is, by this structural interaction, is it inhibits the phosphatase reaction enough so that now the kinase takes over, and so that's why it promotes the kinase reaction at 30 degrees or room temperature, whatever. Okay, so that's that question. Do we have another question?
You may have raised his hand.
Huh?
Walter raised his hand on the.
Well, that was from Walter.
Okay.
Okay, so that was the Q&A, and there's a chat. Oh, that, okay, that's not real. All right, so that's the only one I see so far. Yes,
I'll
Laura.
ask a follow-up question then. So you showed for this stochastic finding of Chi-A that the quantity operates, that's where you saw a difference, and that was phosphorylation dependent. I'm curious if you also get on rates from the A.
Yeah, so that's a great question, and actually one of the reviewers asked us that question. That's kind of, that's a really hard thing to do, and I'm trying to remember why it's a hard thing to do. Because what we would have to do is some of the Chi-Cs may be oriented a little bit so that they're not really all available. So we can't really look at the C as a whole thing in that case. And so what we did is we looked at ones that interacted once so that we know they interact, and then asked the question of what does it matter how long before it will interact again. And in the fairly short period of time that each of these assays were done, you know, maybe 5 minutes or 10 minutes or something like that, that's a hard thing to do. But they did do that analysis, and I don't remember the answer to the question, but I think that there wasn't a big effect on the phosphorylation dephosphorylation status in terms of the initial interaction, but there was this big difference in the dwell time after they interact.
I mean, the reason I asked is because that's a bimolecular reaction, right? So it would be really concentration dependent on Chi-A probably, which, if I understand correctly, right, the system is buffered against concentration changes, so it's kind of curious.
Right, and so that's why this phenomenon is important, because there is a range of concentrations in which the whole thing oscillates, which was based on, when the Columbia lab originally did this, they already had kind of quantified the intracellular concentrations of the proteins. So when they reconstituted the in vitro, they got close to that. But still, just as I said, things are going to be fluctuating, cell division happens, you know, metabolic changes, or things like photosynthesis is happening or it's not happening, things like that. So the system needs to be able to work pretty well within a range of these stoichiometries, and that's what's kind of cool about this phenomenon, because it actually broadens that range by allowing the fast hexamers and the slow hexamers to kind of come back together as they approach the peak. Okay, with the assumption that the longer you dwell on Chi-C, the more likely it is that you're going to stimulate the phosphorylation reaction. Oh, I'm sorry. Oh, what is it? Thank you for your wonderful talk. Really enjoyed it. Did you study if the temperature-dependent phosphorylation, dephosphorylation are just chemical or enzymatic? If the protein is just stored in a buffer and these reactions occur, then what is the source of phosphate? Okay, the source of the phosphate is ATP, okay? If the protein is just stored in a buffer and these reactions occur, if it's just stored in a buffer, well, I'm not sure what you mean by it. If you mean a buffer without ATP, basically they'll just dephosphorylate and just stay there. So I hope that answers your question. You say if the temperature are just chemical or enzymatic. Gosh, I mean, the protein's got to be there. I've always just assumed it was enzymatic. so I don't think that ATP is going to do it by itself unless I'm missing the tenor of your question. So,
You have another question?
Is the dephosphorylation of the two sites sequential like the phosphorylation? Yes, and I said that, but I said it really quickly. Basically, the threonine phosphorylates first, then the serine. And then in the dephosphorylation reaction, the threonine dephosphorylates first, and then the serine.
So I have a question. So it's clear that I think it's pretty important for human health, our human health, is circadian rhythm. I was wondering if there are any phenotypes or diseases that have been associated with genetic mutations.
Yes, okay, so in the clock field, so in case everybody didn't get it, Gary's asking, are there any mutations or diseases that have been associated with clock perturbation? So, for example, you know, a lot of people suffer from what's called delayed sleep phase syndrome, where they really can't go to bed until later, and then they sleep really late in the morning and whatnot. There's some cases of advanced sleep phase syndrome where people fall asleep at 8 o'clock in the evening, even though they don't want to, and wake up at 4 o'clock in the morning. Those are things that are socially kind of problematic, okay? And that there are SNPs, genetic polymorphisms, that are associated with each of those things, okay? There's an association now between obesity and what we call social jet lag. So social jet lag it's not that you're actually flying anywhere, it's that during the week, we get up at 7 o'clock in the morning because we have a job, or we have to go have a class, something like that, blah, blah, blah. But then when we get to the weekend, we go to bed a lot later and we sleep until 2 o'clock in the afternoon or something like that. And so on the weekends, we're really phase delayed, but then the week we force ourselves to come back. So that's what's been called jet lag, social jet lag, because it's the society that's causing us to kind of go through a jet lag type situation. And that's been associated with obesity. And then, of course, shift work is also associated with obesity. So shift work is kind of something that you're kind of dinging the circadian clock. I mentioned seasonal affective disorder is something that's also related to circadian clocks because phototherapy that phase shifts your clock tends to be very effective in that case. And so those are some of the things that just as I'm blathering along that just spring to my mind.
I recall Big Pharma for a while, they were interested in this as a, improving human life, life. And so it's supposed to be, there's some idea that it could extend life, is that right? Extend life?
That could extend life?
Extend your life, yeah.
Okay, so Gary's asking about Big Pharma being interested. I think Big Pharma is interested because if you could really get a jet lag pill and a shift work pill, that would impinge upon a lot of people, okay? Maybe not chronically, but episodically, it would impinge a lot of people. There might be a real market for that. And you said...
Sometimes, resveratrol is the famous story, I think,
yeah, right.
that there was a big pot buy by GSK, and I think the idea there was that they thought that you could find a medication to really extend human life.
Oh, right. Yeah, so could it extend human life? Well, there have been studies in mice where they subjected them to phase shifting every week or every 3 or 4 days or something like that, and they looked at the longevity, okay? And if you start out with kind of older mice, it works really well. If you start with young mice, it's not such a big deal, okay? Because younger animals are just more resilient. But if you start with older mice, if they're forced to phase advance, like they're flying to Europe so that you have to get up earlier, if they're forced to phase advance, the mortality, the survival rate really just is awful, okay? If you're allowed to delay, it's not so bad, okay? But phase advancing, like you're flying east, is definitely a problem for longevity. And so it could well be, there have been other things where, other studies I'm not sure I believe, but there's a study out there that flight attendants and pilots that are subjected to very frequent phase shifting, that the temporal lobes of their brain shrink, which doesn't sound good. And so that frequent phase shifting and disrupting the clock could be a problem for your health and maybe even your mortality. Yes. Yes, ma'am.
So why a nap is so restorative?
Why is a nap so restorative?
Yeah, is it some constellation thing?
Yeah, I mean, it turns out that there's a postprandial effect, okay, for example, in the afternoon, that after you eat, your metabolism just wants to kind of divert a lot of your blood, okay, so there's a physiological thing going on. But it's also true that in human isolation studies, where people have been isolated for months and looked at their circadian rhythms, that many subjects also have kind of a preferred little nap time. So a lot of people, and I just had a lecture in my clocks class from a sleep specialist, Dr. Beth Mallow. A lot of people think that if you enjoy a restorative nap in the afternoon, as long as it's fairly short, so that it doesn't impinge upon your nocturnal sleep, that may actually be a healthy thing, may be good for you. And in some cultures, like Latin cultures or Mediterranean cultures, a lot of times they actually incorporate that in society. They eat a big meal at lunchtime, they take a nap, and then they come back and they work till 8 p.m. I mean, it's probably healthy.
Yeah. When I was in China, we have 3 hours sleep during the daytime. Summer is 3 hours, in the winter it's 2.5 hours. So until 11 o'clock in the evening, you are not very tired.
Right.
So now right now.
So not everybody can hear you, but you're saying that in China, there's some places where you do take a 3-hour break or something after lunch, and then you don't feel sleepy until 11 p.m. or something like that.
Yeah.
Yeah, right. There are a lot of anecdotes like that, and then, the older I get, the better that sounds. I can tell you. Okay, we done?
We're done.
Thanks,
Very
thanks
much.
for your interest.